Dr. Lauren Lounen, and this is a video about aneuploidy and nondisjunction in meiosis for my students. Aneuploidy means that an organism or a cell does not have the correct chromosome number or count. So an and u, an is a prefix that means not, and u means true. So it's saying not true ploidy. That's what the word can be broken down to. This can happen in individuals as a result of a non-disjunction event that happened during meiosis, or rarely in mitosis, but here we'll focus on meiosis, but a non-disjunction event that happened during meiosis in one of the organism's parents. These sorts of events can happen when there's a chromosome mutation that affects the centromere, but there can also be other causes of nondisjunction, and this is an active area of research. In animals, most aneuploidies are lethal, but we will look at some examples of ones that are not. Plants and fungi, on the other hand, tolerate aneuploidy very well. This table shows a number of aneuploid conditions that are very well tolerated in human population, or at least tolerated to varying degrees. Perhaps most famously, or best known, is aneuploidy of chromosome number 21, wherein there are three copies of chromosome 21, and that condition is referred to as Down syndrome. And this is the frequency of that condition at birth in the global population of humans. Trisomies 13 and 18, which are called Pitot syndrome and Edwards syndrome, are also relatively well tolerated, although individuals with those conditions tend to be less healthy and well than people with Down syndrome overall. We tolerate as humans a range of numbers in our sex chromosomes, X's or Y's, relatively well. And there are a number of known conditions. For example, Turner syndrome means that you have only one copy of the X chromosome. You're genetically female, but you're missing one X chromosome. Trisomy X is a condition just called that, or tetrasomy or pentasomy. All of these are actually well tolerated, and individuals with these genetic conditions do pretty well overall. In males, you can have trisomy of the sex chromosomes overall, wherein you have an extra Y chromosome or an extra X chromosome, or there's a condition called Klinefelter's where you have two X's and two Y's, and that occurs at about 40 out of 10,000 individuals. More rarely we see tetrasomy. In many people with these sex chromosome related aneuploidies, the individual may actually not know until later in life that they have such a condition, and it might be identified because the person has gone in for genetic counseling related to fertility problems, because sometimes having these aneuploid conditions can give rise to problems in your own meiotic events leading to or associated with reduced fertility. So here is a karyotype or karyogram on the left of an individual with Down syndrome. And down here, this circle is highlighting the extra copy of chromosome 21. This was diagnosed or first medically described in 1866 by John Langdon Down, and hence the name Down syndrome. And people with Down syndrome today do much better than they did before because we have a better ability to treat and take care of them, and understand that they have special nutritional needs and social and emotional needs and physical needs. And so people with Down syndrome can live a very long life now. There is a relationship between the age of a mother and the risk of Down syndrome in a child. And this jumps up pretty notably after after the late 30s. So you'll notice here's the risk of having a Down syndrome child. It's pretty small in the early childbearing years, but it goes up over time maternally. And that is thought to be because the primary oocytes are in the ovary right from birth. And if you wait until you're in your late 40s to have offspring, your eggs have been in there for quite a few years, and that can lead to increased problems in completing the meiosis required to make a functional gamete. The term nondisjunction means the chromosomes fail to segregate or separate properly during meiosis, and it leads to aneuploid gametes. Then when those aneuploid gametes participate in fertilization events, you get aneuploid individuals. So nondisjunction events can happen in two ways. They can happen in meiosis 1 or they can happen in meiosis 2. And a nondisjunction happening in meiosis 1 is shown on the left-hand side of this figure. A nondisjunction in meiosis 2 is shown on the right-hand side of this figure. So both of these depict how you could get, or what the possible gametes are due to a nondisjunction event in either meiosis 1 or meiosis 2. So if we start by focusing on the left, this simplified drawing is showing just one pair of chromosomes in a eukaryotic cell, each in its sister chromatid state. So we're heading into meiosis. What should have happened is these homologs should have lined up during prophase, synapsed with one another during prophase 1, and then lined up on the metaphase plate, and then been pulled apart uniformly where one homolog made it to each cell. So you'd have like a red one over here and a blue one over here. But instead what happened is both homologs in their sister chromatid state went to one cell and not the other. And presumably there are other chromosomes in this organism, and so this cell over here, isn't actually completely empty, but did get chromosomes from the other pairs. Now what will happen is, as this cell goes through meiosis 2, you'll have no copies of this original chromosome pair making it into the gametes. So these are aneuploid gametes in that they are missing whatever this chromosome pair is. No copies of that in here. They'll have other chromosomes from the other chromosome pairs, but not this, okay? And then over here, what's happening is this cell will do its best to continue through meiosis 2, and these gametes have an extra copy. So these have one copy too many, and these have none. So all of these gametes are aneuploid gametes. One of the ways that you can spot if this sort of, problem happened, if nondisjunction happened during meiosis 1, is that any alleles that are on this chromosome pair, if you had a circumstance where you had like a heterozygote, you would see both alleles present in the gamete. In contrast, over here on the right-hand side of the figure, if the nondisjunction event happens in meiosis 2, so it's happening over here, then what you will see is you'll see two types of gametes that are aneuploid. One is missing a copy of the relevant chromosome pair, and the other has too many, and they are identical. So they would be identical allelically. So in this circumstance, you actually do get two normal gametes produced. So, you have only a 50% chance of having an aneuploid gamete make it into a fertilization event when the nondisjunction happens in meiosis 2, whereas the outcome from a nondisjunction event in meiosis 1 is all abnormal or aneuploid gametes, a little more severe. So there is an additional cause of Down syndrome that I should mention, and it is something called a Robertsonian translocation. Down syndrome means you have an extra copy of chromosome 21, but it can be that you have an extra copy or you have extra amounts of the content from chromosome 21. And this can be due to a translocation event. And this figure is showing that. So here we have chromosome pair 21, and we have chromosome pair 14. And a break has happened between one of the chromosomes in pair 21 and one of the chromosomes in pair 14. A break happened, the cell fixed the break, but it got it a little bit wrong. And what happened is part of chromosome 21 moved over and fused with chromosome 14. So you've got one normal copy of 14, one normal copy of 21, and then this long fused 14-21 hybrid chromosome, and a little chunk that is usually lost as meiosis continues because it's so small, the spindle fibers don't attach to it, and it essentially just kind of goes away. Now this cell is what is called a translocation carrier. It is genetically different And so when this cell undergoes meiosis, the pairing options that can happen can get a little bit messed up or different from normal. And so when this cell undergoes meiosis, there are 4 possible gamete types that can occur. This one is a normal gamete, except for it's lacking any of chromosome 21, so it is not normal in that if it participates in a fusion event, as shown here, this being the gamete from a completely normal meiotic event, 121 and 114, you've got 2 copies of 14, as you should, and only 1 of chromosome pair 21. That's not well tolerated. This is a lethal situation, so we don't see individuals with this aneuploid condition. Here we've got a gamete where we've got 1 copy of 21 and 1 of 14. This is totally normal. This is a normal healthy gamete. And when it fuses with a normal healthy gamete from the other parent, you get a normal healthy individual that can't pass on any sort of aneuploid conditions and is fully phenotypically normal in this regard. These 2 circumstances are also problematic. So here we've got the fusion or hybrid chromosome within the gamete, and here we have the normal gamete. When they fuse, you have an individual that might be mostly phenotypically normal, and they are what is called a translocation carrier. So they have the chance of passing on gametes that could give rise to either non-viable offspring or Down syndrome offspring. In this circumstance, you will always end up with Down syndrome, this circumstance here on the left-hand side, because this gamete has the fusion gamete plus a 21 that is normal. And so it brings all that genetic content in. Now we've got 1, 2, and most of another chromosome 21. That's an extra copy. So while not being quite a trisomy, it's enough extra chromosome 21 that this individual has Down syndrome. And that concludes this presentation.